Good morning, grade 8. We are now in the continuation of our topic, cell division and genetics. We are still in this spermatogenesis and oogenesis. When it says spermatogenesis, it is actually the formation of sperms. Okay, this is actually the formation of sperms, which started with spermatogonium, that's the singular, spermatogonia, plural. So in this cell, sperms are actually created. Okay, sperms are created through meiotic cell division, meiosis. Again, from one parent cell, it will become, or the product are four daughter cells. Sure, how about the egg, formation of egg? Okay, the formation of egg is called oogenesis. Okay, it started with the parent cell oogonium, or that's the singular, plural is oogonia. Okay, now the product of oogenesis is actually one ovum. This is the reason why um the female has monthly period. Okay, this is in the formation, in particular with the formation of egg or ovum. Okay, what happened to the three? Okay, three ootids. Okay, the three ootids will actually disintegrate and becomes polar bodies. Okay, every month a female body creates a healthy ovum or egg. So that's the reason behind. Why do we need to have a single egg every month or every cycle? We call that one as cycle. Simply because the egg serves as what? The egg serves as a facility for the developing embryo. This is where the embryo develops and attach to the uterus with, of course, the placenta. Okay. Let's move now to chromosome structure. Let's take a look at the structure of the chromosome. Okay. Chromosomes are organized structures located in the nucleus of the cell. Okay. They carry information essential to cellular functions such as cell division and cell differentiation. They are actually what carries essential information, okay, as to the cellular functions and cellular differentiation. The term chromosome comes from the word chroma, which means color, and soma, which means body. Chromosomes are called such because they are strongly stained by some colorful dyes used in research. We cannot actually see the actual chromosome if it is not dyed or stained. Okay, it needs to be stained. And of course, with a powerful microscope. They are highly transparent. That's the reason behind. Chromosomes are double-stranded in the nucleus of the cells before cell division. Each strand is called chromatid or sister chromatids. Is made up of long DNA molecule coiled around protein called histones. Okay, the DNA molecule wrapped around protein core called nucleosomes or nucleosome. The nucleosome is the basic unit of chromosomes. Let's take a look at this one later. Chromatids are linked at point called centromere. It is actually located at the center, which divide the chromosomes into two sections, okay, or arms. The short section of chromosome is called the P arm, and the long section, the chromosome, is called the Q arm. So this is now the chromosome structure. Okay, this is the P arm. This is the Q arm. Centromere at the center. This is where the centrioles attach the spindle fibers for cell division. This is where, when does the centrioles attach the spindle fibers? In the metaphase. Okay, it resembles In prophase, okay, the centrioles actually travels on separate ends or opposite ends. Okay. In metaphase, okay, it actually pulls the chromosomes at the center or equator. Okay, you can actually see the spindle fibers attaching to the centromere of these chromosomes in the metaphase. In the anaphase, you can actually see the pulling of centrioles to the spindle fibers, pulling of centrioles in opposite directions. And in telophase, you can actually see cleavage until cytokinesis happens, or the separation of two cells, new cells, or two daughter cells. That's in the mitosis. DNA structure and function. DNA is made up of many nucleotide molecules linked in a long chain. Okay, the basic unit of DNA is called nucleotide. Each nucleotide is made up of the following components. We have 5 carbon sugar called deoxyribose. That's why it is actually called DNA, because it is deoxyribonucleic acid. It's a 5 carbon sugar called deoxyribose. A phosphate group. Okay, phosphate group is actually located in the bone of, or in the base of the DNA, and the nitrogenous base pairs. This is the adenine, thymine, guanine, cytosine. Okay, the nitrogen-containing bases are grouped into purine, which include guanine and adenine, or A and G, and the pyrimidines or pyrimidines, which include cytosine and thymine. Okay. The DNA molecules made up of 2 complementary polynucleotides. They are said to be complementary because the nitrogen-containing bases of one chain are bonded to those of other chain according to the rule of base pairing. Okay, adenine is always paired with thymine, while cytosine is always paired with guanine. A is to T, T is to A, C is to G, G is to C. Hence, adenine and thymine are complementary bases. Cytosine and guanine are also complementary bases. Okay, so as you can see here, we have here the illustration of the DNA. As you can see, it is actually paired. Guanine and cytosine has 3 hydrogen bonds, while adenine and thymine has 2 hydrogen bonds. Okay, this is the phosphate group, which is in the base of the DNA. Okay, and the nucleotide. Let's take a look at the gene structure. Do you have any questions so far? Oh, wait. I can only see Kalina here and Keisha. Nino is out? Okay. Let's take a look at now the gene structure. Okay. A gene consists of 2 polynucleotide chains. One chain is called the template and contains a sequence of nucleotides or bases which help determine the kind of protein produced in the cell. The instruction needed to produce one particular amino acid is considered as the basic unit of proteins, is carried by the specific sequence of 3 nitrogen-containing bases, known as codon. So each codon signifies a particular protein that will be created, that will be generated all throughout the chain of genes. Okay. The information contained in the genes is called the genetic code. All throughout the genetic code is actually paired, or actually sequenced into 3 nitrogen-containing bases, which determines a particular protein to be created. Okay. Let's take a look at the non-Mendelian patterns of inheritance. Okay, according to Gregor Mendel, he is actually the father of modern genetics. He is a priest which studied peas And according to his study, he actually saw some patterns. This is now the Mendelian patterns. But there is a non-Mendelian patterns of inheritance. What are these? Okay. According to Gregor Mendel, the genotype means genome or the genes of an individual organisms. This is the complete set of genes, consists of structures called factors, which are now called genes. These factors control the inheritance of characters and are present in pairs of contrasting characters, okay, or characteristics. These factors separate or segregate during the formation of gametes, the meiosis. Hence, each gamete receives only one factor from every pair of factors. They're independently passed on from one generation to another, and the inheritance of one character does not influence that of the other. Okay? Take note. During meiosis, there is what? There is a segregation, okay, of factors or genes. Okay? They are actually sharing what? Sharing genetic characteristics. So every gamete is unique. Every gamete is actually carrying a specific genetic characteristic, and it is actually what? It is unique. It is. However, scientists observe that some characters do not segregate according to Mendelian's principle. The expression of some characters can be altered by influence of other factors. Okay? By these observations lead scientists to formulate principles of heredity that are not Mendelian in nature. This is now the non-Mendelian inheritance. Okay? One non-Mendelian inheritance is the incomplete dominance. When say incomplete dominance, it is a pattern of inheritance in which one allele for a specific character is completely dominant over the other allele, resulting to another phenotype in which expressed in physical character in combination dominant and recessive phenotype. Hold on for a while. Right? Let's continue. Okay. So again, the incomplete dominance is a pattern of inheritance in which one allele for a specific character is completely or incompletely dominant over the allele of the other, resulting to another phenotype in which the expressed physical character, okay, is a combination of a dominant and recessive phenotype. So this is now your what? A mixture of genes. Like for example, you are actually tall, but you are carrying a gene for short. That's incomplete dominance. Why? You are not expressing the gene for short, but you are expressing the gene for tall. It's incomplete dominance. Okay, the one dominant, the other allele or genes is dominant over the other allele. Okay, let's take a look at this one. Genotype. Genotype is an organism's complete set of heritable genes or genes that can be passed down from parents to offspring. The complete set of genes are called genotypes. Okay. Phenotype. Okay. Refers to the observable physical properties of organisms. These include the organism's appearance, development, and behavior. What can you see that is actually the phenotype? Like, for example, tall, okay, brown eyes, black hair, okay, skin color is brown or white. So those are the phenotype. Phenotype of a specific genome or genotype. Okay. If we are talking about the genes for tall, that is actually under genotype. Tall can be heterozygous tall, which carries a gene for short, or a homozygous tall, which carries only a gene for tall. Okay, but if you're going to check the phenotype, it is still tall, whether it is heterozygous or homozygous. Let's take a look at homozygous. When you say homozygous, homo means one. Okay, it is a genetic condition where an individual inherits the same alleles for a particular gene from both parents. It is considered as pure. Okay, hence the word pure breed. Pure. Okay, so you are not carrying a recessive allele. You're just carrying a dominant allele, pure allele. So hence the expression, if it is tall, all capital T or tall. So you're not carrying a gene for short. How about heterozygous? Heterozygous meaning having two different alleles of a particular gene or genes or mixed. Okay. Hence the expression capital letter T and small letter t. Okay. Capital letter T means you are carrying a dominant allele for tall, but you are actually carrying also a recessive allele for short. So that is actually, if you're going to look at it in the perspective of genotype, okay, it is tall with short. Okay, you're carrying a gene for tall and a dominant gene for tall and recessive allele or recessive gene for short. But if you're going to look at that one in the perspective of phenotype or the physical attributes, you can only see that the individual or the organism is only tall. Okay, whether it is heterozygous or homozygous. Punnett square. Okay, it is a square diagram that is used to predict the genotypes of particular cross or breeding experiment. It is named after Reginald C. Punnett, who devised the approach. Okay, the diagram is used by biologists to determine the probability of an offspring having a particular genotype. This is probability. Okay, if it is probability, meaning to say that it can be in a ratio or it can be in a percentage. So the answers for the Punnett square can be in ratio or can be in percentage, since this is probability. Okay. Punnett square is a tabular summary of possible combinations of maternal alleles with paternal alleles. These tables can be used to examine the genotypical outcome probabilities of the offspring from a single trait allele or when crossing multiple traits from the parent. Okay, this is the tool you use to determine, for us to determine a probable, since this is probability, probable offspring of a particular parents. Okay. Codominance. Let's take a look at codominance. If we have incomplete dominance, we have also codominance. It is a condition of inheritance in which two different alleles for genetic characteristic are equally expressed. Okay? Generally, each individual receives one version of a gene called allele from each parent. So, if incomplete dominance, okay, the recessive allele is actually covered by the dominant allele. For the codominance, it's equal. So it's 100%. Okay, the the distribution of genes is 100% to the offspring. Okay, the dominant allele is being expressed, and of course the recessive allele also is being carried or expressed in the progeny. That's codominance. Okay, if alleles of heterozygous or different, if alleles are heterozygous or different, the dominant allele is generally expressed while the expression of the other allele, which is recessive, is concealed. Okay, concealed meaning covered up. However, in codominance, neither allele is recessive and the phenotype or both alleles are expressed. Okay, I'll be discussing this one moreover in the succeeding slides. Sex determination women in humans, 6 chromosomes are classified as X and Y. Okay. A human being is composed of 23 pairs of chromosomes, 23 pairs. Okay. How many chromosomes do we have? If we have 23 pairs, how many chromosomes do we have? All in all?
36.
40. Come again, Kisha?
46.
46. Okay, correct. 46. Okay, because the 23 of it came from the sperm or male gametes, and the other 23 of it came from the female gametes or egg. Okay, 22 pairs of chromosomes are called autosomes. This is considered as the non-sex determining chromosome, and it is the one responsible for your physical attributes. And one pair of gonosome, which is the sex determinant chromosome. When we say sex determinant chromosome, it actually what? It actually determines whether the organism is male or female based on X and Y. When you say sex determinant chromosome, this will give us an indication that the organism could be a female or male. Okay. If an organism is female, it is actually what? Classified as XX. Okay. While if an organism is male, it is classified as XY. Okay. Again, XX for female, XY for male. Okay. Again, autosomes is a non-determinant chromosome, and it is actually responsible for your physical attributes, while the one pair of gonosome is a sex-determinant chromosome. Okay, this will determine whether the organism is male or female, XX or XY. Sex chromosomes are distinct chromosomes that are involved in determining the sex of an organism. Okay, females have 2 chromosomes, XX, whereas males have 1 X chromosome and 1 Y chromosome, hence XY. Okay, during meiosis, the male XY sex chromosome pairs with separate gametes, sperms. Half of the male gametes contains the X chromosome, and the other half contains the Y chromosome. Hence, again, it separates, okay, to X and to Y, right? Since females have 2 X chromosome, all female gametes or eggs have 1 X chromosome, okay? An egg fertilized by an X-carrying sperm develops into female, whereas an egg fertilized by a Y-carrying sperm will develop into a male or XY. Okay? Hence the sex determination in humans. So again, eggs are only carrying XX, while male is carrying X and Y sperm. So if it actually resembles or pairs in with the egg, it will create a male XY or a female. As you can see there in the figure, in the diagram, you can see 50-50 chances. Okay, 50-50 chances. Okay, or 50 is to 50 or 2:2. Or 50% is to 50%. Sex-linked genes. There are some sex-linked genes. Wait, okay. There are a lot of sex-linked genes, okay? We'll be discussing this one by next meeting, hopefully, since I need more time to discuss this further, because we are now routing to monohybrid and dihybrid process, and I will give a lot of examples with monohybrid and dihybrid process. Do you have any questions so far? Questions so far? Grade 8? By the way, this is the coverage for your exam, okay? For your master test. At the same time, this is the concept also of your project, your learning resource material. Just pick up some of the bits of information here in the lesson itself. Okay, there are a lot of science concepts here, and all you have to do is to create an output. It could be in 3D, or it could be a diorama. I already given you some of the samples, examples in your GC, so that it will guide you all throughout. Aside from that, you can also create a digital output, but then again, the physical output has the biggest grade compared to the digital output. Do you have any questions so far? Questions? Keisha and Kalina? Question, Chair, how long? Okay, that's a good question. Okay, it's a minimum of 5 minutes to 10. 5 minutes up, rather. 5 minutes up. So it should explain the concept. And again, it could be in a vlog or a documentary, narrative documentary. Should be narrated by you. Any other question? Any other question? Okay. I think that will go for today, class. Thank you for your participation, Kalina and Keisha. Have a nice day, everyone. Take care. Yes, Keisha, you're raising your hand. Keisha, did you raise your hand? Keisha? Is it all right? The video could be animated? Yes. Depending on your own creativity, it's okay. I'll still accept that one. Okay? It's okay. It can be animated. Just let me know. Okay? And make it more creative. Right? I think that's all for today, class. Thank you for your participation. Grade 8, Kalina and Keisha. Have a nice day. Everyone, take care. Bye-bye. Enjoy the rest of the day.